Oye, Diego, ¿cómo puedo decir que la sesión de Public Journal Club está ocurriendo ahorita? Running up now is live on now is
Is live now.
live now. Okay. Gracias.
Oigan, yo creo que hubo una confusión horrible con los horarios porque el cartel que a mí me mandaron para publicar en la página web decía que iba a ser una hora después.
Sí, es lo que dijo Eugenio el otro día en el chat, que no sé quién lo quiso sabotear y que por eso cambió la hora intencionalmente en el cartel, en el primero.
Y ya nunca encontré el otro cartel.
Sí,
Pero es que está todo en Twitter.
ay, pues sí, pero la gente no anda al pendiente del Twitter. No todos.
Güey, pero en Twitter además las imágenes pierden resolución. Bueno, pero en las buenas noticias tenemos el mismo éxito en YouTube que el éxito que estamos teniendo en la reunión en vivo. El mismo número de participantes se están viendo en YouTube que ahorita.
¿Ya estamos en live? ¿En YouTube?
Sí, ya estamos en live.
Y nosotros hablando.
Okay, so Victor is in waiting room.
Oigan, entonces con esa nueva estrategia de que ya no podemos enviar los 300 correos, nuestro buen amigo Hidalgo ya no recibió su invitación.
Si no se registró, ya no recibió invitación.
Ya me los están rebotando. Bueno, me rebotó uno, que es el de Sato, Riora Sato. Dice que no encuentra esa dirección de Gmail, pinche japonés.
Ya te
Proporciona
están
una dirección de
uniendo.
correo incorrecta.
Oigan, ¿se escuchan mis notificaciones? Hola chicos, bienvenidos. Welcome to this Public Journal Club session. We will be starting in a few minutes. Hello, everyone. Welcome to this Public Journal Club session. We will be starting in a few minutes. Hello, guys. Thank you for joining to the Journal Club. Today, well, before starting, I'd like to talk about this project. Public Journal Club is a project that we started a few months ago, and in which we are trying to discuss novel articles, novel research papers that related to immunology. And the interesting part of this project is that after the paper presentation and discussion among us, we invite the first author or sometimes even the last author of the work to participate, and we have a question and answer sessions with them. So it's very interesting to hear what they have to say about their own work. Um, So we will be starting in 2 minutes approximately. We're waiting for people to join. Um, so yeah, thank you for joining. Okay, so once again, welcome everyone to this Public Journal Club session. Today, we will be discussing this paper that was published recently on Nature, which is titled C9, the chromosome 9 open reading frame 72 suppresses systemic and neural inflammation induced by gut bacteria. This work was made by, was done in two institutes, the Harvard Stem Cell Institute and the Broad Institute, both in Boston, Massachusetts, and in the Kevin Egan lab, and most of the experiments were performed by Aaron Burberry, who will be joining at the end of this paper discussion to answer some of our questions that we have in this discussion. So just to start, the chromosome 9 open reading frame 72, it's a very interesting gene. It encodes for a protein which is mainly expressed in motor neurons of the spinal cord and the cerebral cortex, and it participates in the vesicular transport of the RNA regulation. It's principally works in endolysosomal pathways, as we will see, and it has a region which has a hexanucleotide repeat, which is 4 guanines and 2 cytosine. And it's the most common genetic variant that contributes to amyotrophic lateral sclerosis and frontotemporal dementia. So usually when people can have some of these hexanucleotide repeats, but if this is over 30 repeats, people can be prone to these diseases. However, even though the people have this repeat expansion, there's an incomplete penetrance in families. So not all of the families will develop amyotrophic lateral sclerosis or frontotemporal dementia. And also the development of one of the diseases or the other depends on the family. So it's very interesting, and it hasn't been explained why all of this happens. Any question about this gene? If not, I would like to also say that this chromosome 9 open reading frame 72 is it has been widely studied using mouse models. However, even there's a lot of variance in the results. So the survival of the loss of function mutant mice varies markedly between reports. So for example, here I'm showing you the supplementary figure 1 of the paper, which shows how in different studies, some of them, from the same author, using different mouse lines, you can see a different trend in the survival. So for example, usually like the controls, the wild type mice survive more than 400 days. However, the homozygous mutants tend to have a lower life expectancy, and they start dying around 200 days. The difference is that, for example, here in figure A, you can see that most of animals die by 400 days. However, only less than half of these animals die, and here around a bit more than a half. And in other publications, there's even no difference between the wild type and homozygous animals. So that's a very interesting phenotype, and it varies depending on the publication. So in here, varies in the publication and also on the place, as we will see. So the first thing they did to try to understand why there's such a high variance on the phenotype of these mice, they divided the mice, and they breed the colony into institutes. So they had the colony in Harvard, and they continue breeding it. And during the paper, it will be known as C9 Harvard, and in the and they also took an they also made an aseptic embryo ready reversion from the animals in Harvard and start breeding them in the Broad Institute. And they checked for the different phenotypes that we will see. So we will here you can see that the two different survival curves, and you see that in Harvard the animals start dying at around a bit more than 200 days, and they get to 63% of survival if they have the mutation. However, in the Broad Institute, only 2 animals died, and at 400 days, which is quite a difference. And that's very interesting because the genetic is the same, and they are both housed in aseptic conditions. However, the difference is that there's a difference in the phenotype of these mice. So also they checked for some markers of immune activation, such as spleen weight, the number of neutrophils in blood, the number of platelets in blood, and the anti-double-stranded DNA as a mark of autoimmunity. And as you can see here, there's a difference, although both of the animal in here, for example, in the spleen weight, the mutant animals in both institutes develop a higher, have a bigger spleen. There's a significant difference, and the Harvard breed mice have bigger spleens compared to the Broad mutant mice. Also, it's interesting to check that there's the phenotype in the neutrophil number and the platelet count that is present in Harvard, in the Harvard breed mice. It's lost in the Broad Institute. So it shows that in the Broad Institute, there's not much immune activation. And although there's markers of autoimmunity, again, there's a difference between the mutant animals bred in Harvard towards those ones bred in the Broad Institute. So yeah, so here, as you can see, there's a difference in the environment, and you can divide it as a pro-survival environment and pro-inflammatory environment that is ameliorating the underlying inflammatory and autoimmune disease found in the mutant mice. Any questions here? I can read you in the chat, or you can open your microphone.
Yes, so I have a question. So basically, the only difference that they found is that these 4 parameters, but I don't know if they check about if both animals reach the same level of disease regarding amyotrophic lateral sclerosis or frontotemporal dementia. So if they check for those other parameters in Harvard and Broad, if they see any difference.
It's interesting that because they don't have I think they have a supplemental figure when they try to show that, but they are only measuring their survival. I think both of the colonies develop symptoms at 28 days after they are born. I think there's I don't know about the difference in the phenotype. That's a very interesting question. So, okay, so we can continue. So they suspected that
Sorry, Diego, I have a question. How was the transportation of the mice from Harvard to Broad Institute? Like I understand that they freeze mouse embryo, and then they grow new colonies from that mouse embryo in the Broad Institute.
Yeah, exactly.
Okay.
Yeah, so the idea is to make it aseptic, so there's no, you're only transferring you're only keeping the genetic part and not the environmental part. And that's very interesting for the next result, because if the genetics are the same and you're seeing a different phenotype, the next thing to check is the environment, and checking for um, the vivarium reports on microbiota. They realize that there are differences in the microbiota from both animal facilities. So what they did next is to, after the mice were born, they start a lifelong treatment with antibiotics or vehicle as control, check for disease onset, and check if the disease and the blood parameters they were checking in the previous figure are maintained or they are changed due to a lack of microbiota. So here in B, you can see that there's at 4 weeks, they are checking the microbiota, and they check the microbiota first, and there's no much difference. But when they after 8 weeks, in which they start, which is 4 weeks after they start the treatment, in the vehicle, there's no much change towards the other 4 weeks, which is expected. But in the antibiotics, all the microbial diversity is completely lost. So it's important to say, I think I will show you later, that the microbial counts are completely lost. Here is just the diversity and the reads, which is just to say that this phenotype, it's worse when you take account the reads, the microbial populations. So again, they check for neutrophils, platelet count, and anti-double-stranded DNA and spin, and all of this in the Harvard group. And very interestingly, when they treat with antibiotics, all the phenotypes that they had seen before, which is an increased number of neutrophils, a decreased number of platelets, an increased activity of anti-double-stranded DNA, and a bigger spleen, are lost with antibiotic treatment, with lifelong treatment on antibiotics, which is very surprising. Because the mice are still with the mutation, but they don't develop any symptoms. It's important to say that these animals also have, due to the continuous treatment with antibiotics, they have problems with the hepatotoxicity, which is expected as the treatment is very aggressive. So what they did also is to check if they could, it's a similar experiment as the other one, but they start the treatment after the disease onset. So not lifelong treatment before the disease onset, but when they had disease, when the moment they started looking at disease symptoms, which is 28 weeks, after the disease onset, they started treatment 4 weeks later with the antibiotics, and they check if there was a change or an improvement in the different parameters. So for example, here you can see that from pre to post, there's a decrease in the in this autoimmunity marker. Also, the neutrophils after 10 weeks with antibiotic treatment are decreased. However, here it's interesting that also in the control, there's also a decrease in the antibiotic treatment with the antibiotic treatment, and the platelet count, it's increased in the group that received antibiotics. So also they check for behavioral parameters. As I said, this mutation affects mainly motor neurons. So if you place the mice in a rota rod, which is just a rotation tool to check for motor coordination. And you check how long the animal takes to fall from that machine. You can see that the animals with antibiotic treatment took longer to fall than the other ones, which is, and they almost stay at the level of the control group, which is very, very interesting. Any questions or comments on this figure or the previous one?
Yeah, I have a observation. In the figures, the effects that are observed, we can see that there is a tendency of the same effect in the wild type versus the mutant. There is the same tendency to how to have an increase
in this figure, sorry, or the previous one?
In this one.
Mm-hmm.
So I think that maybe there's no overall there is no by the microbiota by the affection of the microbiota, but we can see that this affects the antibiotic is affecting also the wild type, no? So it's more like you are affecting all your microbiota, all your microbiome, and this is obviously affecting the immune response that all we know. But the difference between the wild type and mutant mice is by the maybe by the mutated gene, but not is not seen all contribution, especially by the microbiota, no?
Yeah, like it's important to remember that this, although you're changing making a treatment, the mice are still developing symptoms because they have the mutation, right? So there's like also a genetic component on these groups. However, it's interesting to check how just by removing the microbiota, which may seem like a unrelated thing to check, it's affecting the outcome of the disease. So yeah, but it's important to check that it's hard to reach, for example, here in the platelets or the neutrophils, the level of control mice. So any other comments? Why didn't I'm reading in the chat, why didn't they made germ-free animals? Yeah, that's very important. I don't know how it seems like they are just trying to test this, but of course germ-free animals would be the best way to check. However, in those you are also affecting other things. I don't know if there's any publication of C9 open reading frame 72 with germ-free animals. I think that's a very important experiment to do. Yeah, maybe specific pathogen-free animals to find the disease-prone bacteria. Yeah, it's important to say that these animals or the animal facilities were under the limits of the of organizations to they are considered as healthy a healthy environments, and there are no pathogens or described pathogens in any of the animal facilities. However, we will see later that there's a specific Helicobacter species which has been seen to have immunoregulatory effects. It's differently localized in the different animal facilities. So yeah, that's a very good point. which they could use any other approaches to get rid of the antibiotic side effects to check if the phenotype is conserved and if it's really dependent on the microbiota. Also, I'd like to show this figure where they show that this, as you can see here in the vehicle and the vehicle wild type animals and vehicle mutants, there's a difference between the expression of different cytokines and pro-inflammatory cytokines mainly. And it's interesting to see how this cytokine expression is conserved even the antibiotic with the antibiotic treatment. However, there's a difference or there's an improvement in the comparing both mutant animals treated with vehicle or with antibiotics. Again, they don't reach the levels of control animals in any case, so that's important to note. And for continuing checking of the microbiota participation on this phenotype, what they did is to do a fecal transplant, which is changing the microbiota from one animal to the other. So after the disease onset, as in the previous experiment, they start the acute antibiotic treatment. And then after a small antibiotic treatment to deplete all the microbiota, they did the fecal transplant, changing from Harvard to broad feces or vice versa. So here it's not seen correctly, please. In a minute. Sorry for that. So again, they check for the different immune phenotypes they have been analyzing through this paper, which is a marker of autoimmunity and immune activation, the neutrophils and platelets. So they check and see when the Harvard these animals are given Broad Institute feces, they are improving they are decreasing the activity of anti-double-stranded DNA antibodies. The neutrophil count also decreases. However, this count is not as this difference is not as marked as just deleting the microbiota, and this also in the platelet count there's an improvement when changing the microbiota with the in the mutant animals. So I'd like to hear from you any comments in the chat, or if you want to open your microphones on this. So any questions? Okay, so it's I'd like to say that it's important to know that this neutrophil phenotype is not rescued changing the microbiota, which may seem that the previous microbiota has made some damage in the tissue, and that it can also might be changing the permeability of the gut, which they never check in this paper, which would be an interesting question to think about. I'm reading a comment. If there's innate memory or in the precursors, there's an affection. Yeah, both could be possible. I think they lack experiments to really prove or try to assess these questions, but we don't know. So just to say that I show you that signals derived from particular gut bacteria are promoting inflammation and autoimmunity when the there's a problem with the C9 open reading frame 72 function. So then as deleting the microbiota is affecting the outcome and is improving the outcome, they want to check what's really changing in these microbiota or microbial populations. So they took their animal cohorts, and they also took two other ones that they are known. They had the Harvard cohort and the Broad Institute cohort, and they took the Johns Hopkins cohort and the Jackson Laboratory cohort, which both in this Johns Hopkins and Harvard are pro-inflammatory phenotypes, and the Broad Institute and Jackson Laboratory are anti-inflammatory or pro-survival, better say pro-survival phenotypes. And they did a 16S ribosomal DNA sequencing. So they did a principal component analysis, and they checked that both like the four groups are completely different from each other, and they start checking also for diversity in the microbial populations in the samples. So here you can see these are the different samples they took from the Harvard University, the Johns Hopkins Institute, the Broad Institute, and the Jackson Laboratory. As I said, these two are pro-survival and pro-inflammatory, and they are grouped, the microbiota groups as in pro-inflammatory and pro-survival, which means that microbiota is important, or microbial diversity is important in the phenotype of the mice. Also, they check the abundance of different species, and when they did this in the animals and when they did the fecal transplants. And here you can check that their animals have a high proportion of Harvard only microbiota, although the higher proportion is shared microbiota. When they do that, only making the transplant from Harvard fishes given to Harvard animals, they start getting some microbiota, might be just by the procedure. But it's important to check how the Broad Institute, that only has some specific microbiota only in the Broad Institute, when they do the transplant, this microbial species appears in the Harvard animals. And checking for the transplants, it's interesting to see how the Harvard to Harvard transplant is more related to the only Harvard animals, and when they do the transplant, the microbiota resembles more that of the Broad Institute. So it's just to check that the microbial fecal transplant is really affecting the microbiota and the microbial populations.
Diego, I was yes, I was wondering if they that's why was my question about why they didn't like identify the strain of bacteria that was prone to induce disease, so you could only put the bacteria and see if the only adding of the bacteria will induce disease. My second question will be, what would happen if they would mix Harvard feces with Broad feces, like 50-50, and put it in an animal to see whether if there's a strain of bacteria that is more competent against other, so maybe the animal will be rescued. I don't know if having the two at the same point. And my third point will be that, like I said before, like a good control would be like putting the animal in a third place independently of Harvard or Broad Institute facility. I don't know.
Yeah, I think I think they did lots of controls, but they also lacked some. Yeah, I think the way they did the fecal transplant is first deleting the microbiota, because I guess the Harvard microbiota is more fit than the broad microbiota. So they are taking over the broad microbiota, and that's why they couldn't see any difference. So I guess that's why it would be important to ask this to the author to see why he decided to do the experiments this way and not in a cleaner way. We could say. And your first question was, what was it about?
Like 50-50, if you put 50-50 of the Harvard and broad to see what would happen, or identifying the strain of bacteria that was dominant or giving the phenotype. So you could play with the amount of bacteria to see.
Yeah, they make an approach, or like they try to identify the strain of bacteria. The problem is that there are lots of different bacterial species in the Harvard Institute and in the Broad Institute. So it's hard to isolate one and say that, okay, this is the one that is affecting. However, as we will see in the next figure, although the alpha diversity, that is the diversity within as the different samples, is might be changed a bit, and this Broad Institute and Jackson Labs have all like a higher bacterial diversity. And it's important to see that the epsilon proteobacteria, which are Helicobacter species, are only found in the pro-inflammatory or pro-disease facilities. So it's interesting to see they already knew that these Helicobacter species have immune participation, but it's interesting to see how only these species are found in the Harvard or Johns Hopkins. So I think with this figure, they tried to argue that Helicobacter species could be it's like a very clear candidate to be the ones changing giving this phenotype, but they cannot rule out all the other bacteria that they found in the sample. Here in H and I, you will see Helicobacter PCRs, and just to check that the Harvard and Johns Hopkins are again showing these Helicobacter species, but not the Broad Institute and the Jackson Lab. And when they do the fecal transplant that from broad feces to Harvard animals, the Helicobacter species disappear, which again would argue that Helicobacter species are the ones that are giving this phenotype, or at least they are contributing to this phenotype. So in this part, they try to argue that microbiota changes are underlying the phenotypes, and in particular, they try to make the point that Helicobacter is an important bacteria in this process. So finally, they all this disease is marked by motor problems. So they are trying to check if there are problems in the. No, sorry. First of all, they try to check if the what is this bacteria contributing, or how is this how are the myeloid cells of these mice responding to foreign stimuli. So they get bone marrow derived monocytes, and they culture them with different TLR ligands, and they see that they are the TNF alpha production of these cells is increased in the mutant animals. And also, it's when they use the feces from different from the two facilities to activate the bone marrow derived macrophage. It's interesting to see how both feces are inducing the TNF alpha production, but when they do a dilution of these feces, you can see that the Harvard feces are with a lower concentration of of Harvard feces, you can activate and induce TNF alpha production, which shows that the microbiota is directly affecting the cells. So as yep.
Sorry, just in this figure, would you have done like the same experiments using Harvard and Broad Institute mice? Because they only did this experiment with the Harvard mice. So I don't know if maybe, I mean, the microbiota would remodel the bone marrow or the progenitors to produce more proactive or pro-inflammatory myeloid cells. So
yeah,
what would
I think they wouldn't find that much of a difference because they are only if that's the case that you're saying, right? Like if the microbiota is remodeling the progenitors and not only the cells. But I think it's at least it was not the scope of the paper. It would be an interesting result, but I would expect to see that the same, that Harvard theses are also inducing more TNF alpha. But yeah, maybe comparing both animals, you would see again that the Harvard animals are cells are more prone to producing TNF alpha.
Yes, or maybe like doing a bone marrow chimera, like reconstituting the animal with a wild type hematopoietic compartment and see that maybe the cell phenotype is dependent on the myeloid cells interacting with bacteria, or that are deficient of this or have this mutation. So yeah, I would love to see that experiment, but I don't know why they need to do that.
Yeah, I think this paper is like a first observation that microbiota is really changing things. So I think they don't describe really a mechanism in the whole paper. It's more like observations trying to put together to make a coherent story. But I hope they publish more about this to really prove that this is the case. So yeah, finally, they wanted to check for myeloid cell infiltration and myeloid microglia activation in the different animals. So here they are just checking the control animals and the mutant animals. Again, these are only made in Harvard. And it's interesting to see how the animals, like as expected, is just try to show like why is this happening. Like they are using CD45 to check for hematopoietic cells and the infiltration of these cells, and this is only happening in the loss of function animals. And they also check for IgG antibodies, which are autoimmunity markers, and they see this only in the animals are they see this phenotype. Again, it would be interesting, I guess, to check, not comparing wild type to mutant, which has already been shown, or it was thought that this was the case, but comparing both mutant animals from different facilities to see if this was this you could see something similar to this. And here in B, they are just checking by flow cytometry the microglia and myeloid infiltration in both groups when they give vehicle or when they give antibody. So you can check that when they give vehicle, there's a loss in the mutant animals. They lose microglia and there's lots of myeloid infiltration, which might be really directly affecting the microglia. And however, when you give antibiotics in this group of animals, you recover the phenotype, and it resembles more that of the wild type mice. They check also for how where the IgG where it's binding, and this is a marker of this is used to mark microglia in the lumbar cord, and they you can see how the IgG is really colocalizing with the microglia, which it's very interesting, and it's kind of the how it's similar to what we see in the previous figure when because of the infiltration there's less microglia. Here it could be the case that microglia is getting destroyed by the immune cells and the immune infiltration.
Diego, but they didn't show any numbers of microglia. They just show frequency, right?
Yeah, here they show they just show frequencies.
So we don't actually know if there are less microglia not having
Yeah, that's true. That's true because because of the myeloid infiltration increasing, the frequency of microglia should decrease here. That's right. Also, they check for the neutrophil what is this myeloid infiltration or the immune infiltration, and they check that it's mainly of neutrophils and CD4 T cells. The neutrophils in the spinal cord and CD4 T cells in the forebrain, but they don't discuss much about it. And okay, finally, they check for the activation or the activation level of the microglia, and they use DECTIN1 and IBA1 to check for this. And you can see that the activation level in its IBA1 is expressed in both wild type and mutant animals, but the DECTIN1, which is like they use as a marker of activation, it's widely expressed only in the mutant animals. And this is just showing how the treatment with antibiotic is affecting the CD45 cells found in the spinal cord, and how with antibiotic treatment the DECTIN1 expression is decreased. And here you can see the quantification of this. So again, this data shows or tries to argue that the chromosome 9 open reading frame 72 model that they use is favoring peripheral immune cell infiltration to the spinal cord and microglial activation. So we see in this paper that there's an environmental that the environment is changing the phenotype of these mice, and there's like a pro-survival environment can ameliorate the underlying inflammatory and autoimmune disease. That signals derived from particular gut bacteria are promoting inflammation and autoimmunity when the function is diminished, and that the microbiota changes are underlying the phenotypes, and probably Helicobacter species are very important for this, and that the loss of function in this gene is favoring the immune cell infiltration and microglial activation. So I have questions in the chat. Is it possible to improve motor function or prevent sclerosis with that? I guess that's what they are trying to argue. They are trying to see if affecting the microbiota or trying to check for microbial species in humans could affect sclerosis, but only the sclerosis affected for the C9orf72. Like motor function in general, I think it would be hard to imagine that, because in the wild type animals, any treatment is not improving their function. So, but yeah, in case you're a carrier of the mutation, a diet full of probiotics, it's hard to say at this moment, because they are not really saying which species is the one that is affecting. So I guess Helicobacter species are widely expressed among humans, but it's hard to make this connection with this with the information shown in the paper. So any other questions or comments?
Yeah, Diego, I have one.
Yep.
Do you know if there is identified different types of microglia, like macrophages have we have in M1 and M2, or anti-inflammatory and pro-inflammatory macrophages? There is the same for microglia.
There are some subtypes of microglia, but mainly they differ in the location. I actually don't know if they're like within the same location. They are. I think yes, there are different types of cellular phenotypes that all of them are called microglia, but it's like microglia specialized in different functions, like to nurture the neurons or to activate or not. So I guess yeah, there's only a specific population of microglia. To it would be interesting to ask about that, like if different microglia or which is the microglial population that is affected.
Okay, and I have a comment. They don't check. They found that there is a difference in the population of Helicobacter pylori in the two different places of Harvard and Broad Institute. So it will be fine if they or good in the future if maybe they make more insights about if you have mice specific germ free or for Helicobacter mice. If you can recapitulate no the effects in the mutant mice, so it could be interesting if to check if the Helicobacter is indeed the responsible for this
Yeah,
phenotype of plasticity.
here they don't show like the opposite for this, right? Like for example, here they show that the broad fecal transplant to Harvard decreases the Helicobacter, but they don't show if the opposite is true. If the Harvard affecting the broad would be increase the um, Helicobacter species, and you could have problems in these mice. Someone else? So yeah, just to finish, what we see is that there's a pro-inflammatory environment that makes like which are hyperstimulatory microbes that that this in addition to sensitive mutant cells will induce autoimmunity, inflammation, and behavioral abnormalities, and decrease life expectancy and premature death. And that might be caused by immune cell infiltration and microgliosis in the central nervous system and the spinal cord. However, at different environments or changing the microbiota with antibiotics or fecal transplant will make the reduced stimuli, and that will, even though the cells are still sensitive, will this interaction will change, and that will lead only to some inflammation and improved behavior and normal survival. So yeah, that is because there's neuronal homeostasis in these organisms. So if there are no more comments, we are just 5 minutes away from the questions and answer session with Aaron Burberry. So if you have any comments or questions, please feel free to write them in the chat or open your microphone when the author is here.
Chicos, ¿les gustó el paper?
Sí, estaba bueno. Ya.
So we encourage everyone to stay for the question and answer session. It would be very interesting to hear what Aaron Burberry has to say with all these questions that came out in the discussion. So I hope you you have your own and feel free to ask them. It's a very, very good opportunity for you and to practice about like the so to have some criteria when evaluating a paper.
Yeah, very interesting.
Hey guys, I have a question concerning the methodology
Yep.
that they use to measure. They say they measure TNF levels in the supernatant of the macrophage.
Mm-hmm.
But I was wondering, in the paper they said that there are many cytokines that are also elevated. Would you happen to know why did they measure only TNF and not the others, or maybe one or two more cytokines?
Yeah, it's I think it's like I think they did it because it's convenient. TNF alpha is super easy to measure. It's like one of the most released cytokines when you are doing this kind of experiments. And also not all the cytokines are released by these macrophages, but TNF alpha is one of them.
Okay,
So,
thank you. So
okay, so welcome, Aaron. I'll just introduce you. So today it's here with us, Aaron Burberry. Thank you for joining us. Aaron is a graduate student in the was a graduate student in the Gabriel Nunez lab in the Department of Cellular and Molecular Pathology at the University of Michigan. And there he identified modifiers of pattern recognition receptor signaling pathway implicated in the Crohn's disease. He also determined how microbial infection impacts the function and phenotype of hematopoietic stem cells. And now he's presenting this work with this relation with microbiota and neural function. So welcome, Aaron.
Yeah, thanks so much for having me.
Yeah, so guys, if you have questions, please write in the chat numbers. So from 1 to 10 or many questions as you want to start this question and answer session. So Aaron, to begin with, I'd like to ask you, how was, before starting to get into the science, how was doing the experiments in 2 institutes at the same time? Like, how was the scheduling of your days in the lab and all that?
Yeah, this project really spanned years of work in our lab. It was definitely a huge endeavor by many members of the group. So we had been interested in studying this mouse model for a long time, since the discovery of the C9orf72 repeat expansion mutation back in 2011. And we and many other groups have really been trying to understand what contribution, what pathologic mechanisms of that repeat expansion might be contributing to the neurodegenerative diseases, ALS and FTD. So we had studied these loss of function mice. So we made the mutations in the mice and studied them in our Harvard facility for several years. And we characterized some of the phenotypes that developed in that mouse model. And we published a paper back in 2016 that predates the one that you guys journal clubbed, where we basically characterized the autoimmune and inflammatory phenotypes that develop when you get rid of this gene. And it really supported the hypothesis that loss of function of C9orf72 might be contributing to changes in organismal health in these mouse models and potentially in the patients. But really, as you guys read in the paper, some people saw the phenotypes that we did, some other groups that were studying the same mouse model, but many other groups also saw a varying degree of pathology in these mice. Certainly, there were central features of the disorder, the splenomegaly that everybody seemed to recapitulate, but then the extent of the systemic inflammation and autoimmunity seemed to change across time. So really we had this opportunity with our dual affiliation with the Broad Institute and Harvard to take a look at how these mice were, how the disease would develop in mice that were reared at different institutions. And as we laid out in the story, it was remarkably different, particularly with respect to survival and the extent of inflammation. So it's been a really interesting adventure, and as you mentioned, challenging to do studies across 2 different institutions, but really informative in the end.
All right, thank you. So we'll start. I think the first question is Eugenio Contreras. So if you want to ask.
Hi, Aaron. Thank you very much for being here. So, and first of all, congrats for your incredible work. I got a couple of questions. My first question will be maybe methodological, or why I mean, when you analyze the microbiome and the diversity, and you found that Helicobacter was enriched in the Harvard microbiota, why didn't you prove like isolating the Helicobacter and put it back into a broad animal, just a Helicobacter strain, to prove that it was in fact the Helicobacter family of bacteria, the ones that are giving the phenotype?
Yeah, so this is a pretty straightforward follow-up experiment to our paper that I think is warranted. One challenge was that many mouse vivariums do not want these pro-inflammatory microbes present in their animal facilities. These are not pathogens per se, as we mentioned. Wild-type mice with these live normal lifespan, but they do have the capacity to change inflammatory phenotypes. So really, the Broad Institute, and I imagine many other facilities, don't want to knowingly introduce these microbes into their animal facilities with potential consequences. So, to follow up on this point, what we are planning to do is to rederive our C9orf72 mice germ-free. And then we would be able to do a whole host of experiments like the ones you're proposing, where we culture individual microbes and reinstitute them into these germ-free mice and see which ones are able to promote the phenotype or to suppress the phenotype.
Okay. Yes, that was, yeah, I was exactly thinking about the germ-free mice. So why didn't you did the germ-free mice? But I got the answer. So yes, my second question would be, when you analyze when you derive macrophages from the bone marrow, from the knockout, and you see that the knockout produce more TNF alpha when putting with LPS and whatever. I have 2 questions in regarding this. Why didn't you prove or did this experiment using the Harvard mice and the Broad it was just enough with the Harvard, or and my second question would be, if you do like a bone marrow chimera, like transferring the hematopoietic, like a wild type hematopoietic compartment to a knockout mice, would you see the same phenotype?
Yeah, let me try to take these questions one at a time. So as the data that's presented in front of you illustrates, so we took the bone marrow derived macrophages from the mice derived at the pro-inflammatory Harvard facility, and we cultured them, and then we tried to model the potential environmental effects of the fecal preparations from the two facilities. And the first thing that was important for me to notice from these is that in the media only condition, so when these macrophages were cultured for 7 days just in vitro, they did not spontaneously produce TNF alpha. So it suggests that even if they were exposed to an inflammatory environment in vivo and those mice reared at Harvard, that this was not perpetuated in these in vitro settings, at least by 7 days worth of culture. But when we added either these microbial mixtures from the Harvard or the Broad facility, or the synthetic microbial mimetics on the left side of the screen, you can see that the macrophages respond to these pro-inflammatory stimuli by producing the TNF alpha. And then also we saw this interesting distinction between how much of the cytokine was made depending on which mixture of the microbes we added. So one really important control to this experiment on the right was that we used PCR to carefully match the amount of eubacteria that we added to both macrophage preparations. So we made sure that the same number of eubacteria, because these are complex mixtures of feces, were added from the Harvard facility and the Broad facility. And when we did that, we found that the Harvard microbes induced a higher level of TNF alpha production. So on the very far right at that 1 to 5 dilution, you see this kind of environmental difference where the constituency of the microbes in the Harvard feces elicited a larger inflammatory response relative to that when we put the Broad feces in. And then on top of that, just environmental difference, we saw this genotypic difference where if you had a loss of function mutation in C9orf72, you elicited even more cytokine. So these results suggested to us that there were genetic and environmental contributions to the amount of inflammation that was occurring, and a potential mechanism to describe this. And if you look, at even the lower dilutions, the 1 to 25 that we put in this, it was really striking that the only time that we saw really high levels of TNF alpha production induced was when you had the Harvard microflora and the C9orf72 loss of function mutation. So at these lower levels of stimulation, it seems like both of those contributed. The second question, and I can answer questions in response to that, but the second question you asked was about bone marrow transplantations. So in our 2016 Science Translational Medicine paper, we described those studies. So we took mice that had either the C9orf72 loss of function mutation or a wild type genotype, and we did reciprocal bone marrow transplantations into either wild type mice or C9orf72 deficient mice. And what we saw in those series of experiments is that if you put wild type marrow into wild type mice, the mice survive for the extent of the experiment. And if you put mutant bone marrow into mutant recipients, you could kill the mice at a rate, basically the same rate that you would see without these bone marrow and lethal irradiation. When you put wild type marrow into a mutant mouse, you could extend the lifespan of that mouse by about 45 days. And if we looked in the plasma of those, we saw a mitigation of the inflammatory and autoimmune phenotypes that we had characterized previously. And then if you put mutant bone marrow into a wild type mouse, you could kill that mouse and you could give it autoimmunity. So it really supported the hypothesis that the gene C9orf72 was functioning within the hematopoietic system to mitigate these or to mediate these inflammatory and autoimmune phenotypes.
But the bone marrow or the hematopoietic compartment does not affect the phenotype of the mice regarding like sclerosis or motility or something related with the mutation.
Yeah, so we have extended our behavioral characterization of these mice, and we find that they have motor deficits as measured by the rotor rod. And so, we've been able to improve the behavior of these mice, particularly by treating with antibiotics and suppressing this environmental effect. So we do think that the inflammatory state, which we think is largely mediated through the hematopoietic system, we do think that it has an impact on the motor system of these animals.
Thank you very much, Aaron. Congrats.
Thanks.
Right, thank you for those answers. The next question is Caleb and he's writing in the chat that can antibiotics help improve the prognosis of the development of the sclerosis disease, or for the carriers of the mutation, would a diet with specific probiotics improve also prognosis? What do you think on those?
Yeah, so I mean, this is an interesting extension of our findings that would suggest that perhaps antibiotic treatment could be a potential therapeutic for individuals with ALS. So along these lines, I think it's important to mention that antibiotics have been used in ALS patients. There was a ceftriaxone study that was done, and it showed promising results in phase 2, but when it was extended to clinical trial phase 3, it did not show an overall benefit in those patients. And there's a number of reasons why that could be the case. What I would want to propose is that really we need to take a careful look both at the genetics of these individuals with ALS, as well as a deeper understanding of the microflora in those patients. So ALS is a genetically heterogeneous disease with more than 25 different mutations that can lead to familial forms of the disease. In addition to that, the vast majority of ALS patients are so-called sporadic with, in most cases, unclear genetic contributions to the disorder. So what the field thinks, if I can summarize, is that it's possible that each of these different genetic mutations might be contributing to motor neuron degeneration through different pathways. There might be some pathways that different mutations converge on, particularly TDP43 pathology seems like an obvious candidate. But it's likely that one therapy that works for a particular genetic class of patients might not work on all the other genetic conditions of the disease. So to basically put those studies into context, I think it's important that we do future trials knowing whether the patients, or maybe using C9orf72 repeat expansion as an entry criteria, and then profiling the gut microflora of those individuals. So you would like to know before you started treating patients with antibiotics with the hope of suppressing certain microbial species, what the particular bacterial species is within those individuals that is driving the disorder. And we really don't know this yet. We don't have enough information yet in humans about what those potential contributing microbes are. So you'd want this kind of biological readout of whether your therapy is affecting the microbe that you actually, that you wanted. And then, yeah, whether specific probiotics would also improve the prognosis. So again, if we can understand which microbes might be contributing to disease pathogenesis, we might be also able to identify microbes that could reverse it. So could we pair antibiotics by introducing other pro-survival beneficial microbes? And that's certainly something that we're interested in pursuing.
Thank you so much. So Paulina is next.
Hi, my name is Paulina. I'm a biological and pharmaceutical student from Mexico. And you already answered my question. I had a question concerning the pathways that were activated, that if you consider that it was important based on your results to characterize them, to maybe use them as a potential therapy or potential preventive treatment to before the ALS or dementia appeared in the patients. Well, first in the mice, but then if you think that many experiments later, it would be useful for treatment of the patient, of human patients. Do you have plans for doing that?
Yeah, so really our immediate plans are pairing with individuals in the clinic to try to do these sequencing studies. So we can find families that have the C9orf72 repeat expansion, and we can track basically the microflora using these gut sequencing efforts to see what the microbial species are within their guts before they get the disease. Do these change when the individuals convert to neurological disorder? Are there microbes that are associated with that conversion to neurological disease? How does this correlate with their prior inflammatory history? There are case studies that show that ALS individuals and frontotemporal dementia individuals in some cases, we're more likely to have a prior history of autoimmune disease before their neurological condition. So do the microbes within their gut correlate with their prior history of inflammatory or autoimmune disease? And then are there signature of microbes that might be associated with that conversion to disease? And then a necessary follow-up would really be mechanistic studies. So you'd want to take those microbes either individually or as complex communities and put them into models that would allow you to determine whether those microbes are inducing either neurological disease, inflammatory disease, or mixture. So you could kind of break apart the mechanisms.
Yes, because I was thinking in the germ-free mice, it was easier. Well, you can do a better relation bacteria effect than in humans because I think that the environment is a little bit more complex. And if you alter one bacteria species, then you can alter other effects, not necessarily like neuroinflammatory, but maybe in the gut environment of the person or other effects that are not necessarily related to ALS.
Absolutely. I mean, our bodies are very complex and interrelated. So you'd want to understand how the microflora is changing all aspects of your physiology and how that might contribute to eventually neurodegenerative disease.
Thank you.
Thank you. So Susan in the chat asks if did you try to physics like the fecal transplants at equal fractions? Perhaps some microbe dominance exists between the two samples. Thanks, Aaron.
So we didn't do this study, but that is certainly interesting to see if the pro-inflammatory microbes dominate over pro-survival microbes. So we did look I'm trying to think of our results in the context of what we actually did do. So we took mice reared at the Harvard facility and we treated them transiently with antibiotics to deplete the microflora that they had. And then we put in brood feces. So it's not like the germ-free experiment that you're talking about, but it is somewhat of a competitive experiment. We suppressed the pro-inflammatory microbes and we put in the pro-survival microbes. And we saw that I think we said about 75% of the microbes present in the Broad facility re-engrafted into those mice. So it wasn't the case where it's impossible for them to re-engraft in the presence of pro-inflammatory microbes, but we did show that, in particular, Helicobacter was suppressed by that transient antibiotic administration. So we would need to do follow-up experiments to see whether those individual microbes that we think might be having a large effect on the inflammatory milieu might overtake the pro-survival microbes. And it obviously has implications for the studies that, such as like fecal transplants or probiotics. Just my knowledge of this field is that it's generally challenging to get probiotic microflora to engraft an individual if you don't pre-treat them with antibiotics or some other way to clear out the intestinal niche. So, from a clinical standpoint, this is certainly something that you would want to explore once you find the protective microbes that you want to introduce, of ways to introduce them and have them be long-term established.
Thank you. So thank you for that answer. It's very interesting to think about that. Emmanuel is next. Emmanuel, are you there?
Can you hear me now?
Yeah, now
Oh,
we
thank
can.
you. Yeah, thank you, Aaron, for being here. This was a great research work. Well, I am Emmanuel from Mexico too, and I want to know, do you think that microbiota contribution to this, to the phenotype of C9orf72 is exclusively of some species, or is due the interaction of any bacteria with the immune system?
Could you repeat your question again? Sorry, you broke up a little bit.
Do you think that microbiota contribution to the phenotype of establishment, the phenotype of the C9orf72 mutant mice is exclusive of some species of or some species of bacteria, or is due the interaction of all type of bacteria that can stimulate the immune system?
Yeah, so it's basically the question, do we think individual microbes can mediate the phenomenon, or do we think it really takes these communities of microbes to drive the phenotype? And really the answer is that we don't know. We need to do the experiments that I alluded to previously, which is taking the germ-free mice and reintroducing individual microbes to see whether they can drive the inflammatory phenotype. And I think there are examples in the literature that show that individual microbes, such as ones that were present in our Harvard pro-inflammatory facility, can drive autoimmune and systemic inflammatory disorders. So there's certainly literature support that individual microbes can do this by themselves. But in this particular case, we basically just need to do those experiments. So we've mapped out a way that you can do those both in vivo and then also using this in vitro correlate assay with the macrophages to basically see, are the macrophages hyper responsive to one class of microbes or another, and what are the components of that microbial species that are driving that pro-inflammatory response.
Okay, and you were focused only in the good microbiota, but do you think that also infections in another tissues could induce this exacerbated pro-inflammatory phenotype in the C9 mutant?
Yeah, so, as you mentioned in the paper, we focus primarily on the gut, which has the largest community of microflora, but all of our mucosal surfaces have exposure to microbes, our skin, our oral, nasal, and reproductive cavities. So there is certainly a potential for immune cells at each of those interfaces to interact with the microbes and contribute to the systemic inflammation that we see in the mice. And if you look at the causes of death in the C9orf72 loss of function mice, you see that it's really a mixture of different outcomes that can lead to either the spontaneous mortality or the necessary euthanasia to cause these mice to die. Some mice die of dermatitis, some mice have increased immune cells in the lung, increase in the liver. Some mice get changes in their vasculature and internal bleeding. So really we think that inflammation throughout the body is occurring, and it basically could contribute to death in many different ways. But our results in the gut, specifically with orogastric inoculation of the microflora from the Broad facility, would really support that the gut microbe is a large contributor to these phenotypes and can mitigate those if you put different species in.
Okay, and finally, maybe these your results are challenging the famous hygiene hypothesis that many scientists think that the exposure to bacteria or to pathogens is through your life is beneficial about the development of autoimmune disorders, but your results are contrary. You see that when you have this mutation, the exposition to bacteria pathogens maybe is inducing or promoting the development of autoimmune or neuro-related disorders. So, can you talk us more about your perspectives, your future perspectives about this project in this area?
So I would not go as far as to say that our results discount the hygiene hypothesis. I think when thinking about the immune system in general, it's really important to know that, as you guys well know, it's a double-edged sword. So you need the immune system to provide protection against potentially harmful microbes, but you also need the immune system to promote wound healing and to suppress inflammation. And really, it's a delicate balance with many interrelated pathways, both within cells and between cells, that regulate whether you get these excessive inflammation or if you clear inflammation and you suppress it. So I think there are many layers to that question. How does prior exposure to microbes influence the either innate or adaptive immune cells to subsequent re-exposure with either the similar microbial species or different microbial species, and how does our individual exposure overlay with our genotype and our individual susceptibility to having a chronic autoimmune or inflammatory response. So I think it's a really complex and nuanced question that needs to be approached both from the perspective of what your individual genotype is and what your exposures throughout your life through the microflora are.
Thank you, Aaron, for your answer.
Thank you, Aaron. So next in the list is Barbara Hunch, and she's asking if, do you know if humans with this mutation are more reactive to inflammatory signals?
Yeah, so this is an important avenue of research that we and others are pursuing. So taking cells from individuals with the C9orf72 repeat expansion, myeloid cells, either microglia or other peripheral myeloid cells, and then exposing them to microbial components and seeing whether the changes that we see in this mouse model, loss of function, are also recapitulated with the C9orf72 repeat expansion. So that's certainly an important avenue to pursue. And if that was the case, which our results would suggest should be, then you might be able to tease apart what signaling pathways within those myeloid cells are contributing to inflammation, and could they be potential therapeutic targets. So that's an important avenue of research that we're interested in pursuing.
Thank you. I also have some questions myself. I wanted to ask, it was for me very interesting to see how treating these mice, these mutant mice with the microbial components, it's enough to, or the cells of these mice it's enough to start or to have an immune response. So I was wondering if you checked for gut permeability, perhaps, that might be helping to get these components to where the immune cells are, and probably even to the spinal cord.
Yeah, absolutely. That's an important consideration, because there are other models of gut inflammation and even neurodegeneration where permeability of the gut can contribute to excessive inflammation. So we did the studies, which we haven't published yet, but we plan to publish soon, where we fed the mice fitsidextran and then measured how much of that molecule went into the plasma. And we didn't see any differences in the mutant mice. So it doesn't support changes in permeability of the gut to those particular molecules, but because we know that immune cells within the gut constantly survey the microbial components, it still can support a model in which excessive inflammation of gut immune cells could be exacerbated in response to certain microbial communities.
All right, thank you. And the other question was regarding diet. So we know that diet changes can affect microbiota like really quickly and really dramatically. So I was wondering, have you tried changing just the diet in these mice, mutant mice, and you can see changes in the behavioral or immune phenotypes?
So we haven't done those experiments, and I think they're quite interesting to pursue, given the kind of monumental role of the gut microbe in this model. So feeding them a high-fat diet or a diet that would induce short-chain fatty acids, and looking at the metabolic profile in the plasma to see if that was changed by the diet and whether there's a genotypic effect in that. So we haven't done those. I think certainly we could consider them, and other groups should consider them as well.
Okay, thank you so much. Someone has any other question?
Yes, can I have one more question, Aaron? Yeah, it would be, Diego, if you could go back to the figure 4, just because of the question of the numbers.
Oh yeah, if you.
So here, Aaron, my question is regarding if this difference, you only see it in frequency. If you see the same phenotype in frequency and numbers are only in frequency. So maybe the microglia is just being the same in the knockout in the wild type, and just you are changing the infiltration number of the cells. And my second question would be, I was checking that you did this experiment at the 55-week-old mice. So I don't know if you did this in this particular time, or if this phenotype or infiltration is dependent on the age of the animal.
Yeah. So you're correct that the percentages that we describe in the figure that's showing right now was in relation to CD45 positive cells, the percentage of CD45 positive cells. And if you go into the extended data, I believe it's extended data figure 8, we actually use mass cytometry to characterize these populations in more detail. And we took total cell numbers of the spinal cord. And what those results showed, as you alluded to, is that we really didn't see changes in the total number of microglia within the spinal cord of the mice, but we did see significantly elevated levels of neutrophils, which would be indicated by these Ly6C high cells in the figure, as well as CD4 positive and CD8 positive T cells at these aged time points. So we think that it is indeed infiltration of peripheral immune cells into the central nervous system of these mice. And you asked about the temporal nature of this. So in kind of unpublished studies, we've been profiling the central nervous system of these mice at younger ages and at older ages, and we see that it is a progressive response. So it kind of magnifies with time in these mice.
Thank you.
Well, thank you so much, Aaron, for all these insights and answering the questions. Finally, we just have this, we try to give final ask at the end, our author, if you have a take-home message from your paper for the audience.
Yeah, my take-home message would be, if you're doing mouse modeling studies, you should be aware of what microbes are inside your facility. The easiest thing to do would just be to go to the animal health monitoring reports. Every animal facility has to do these. They look at sentinel reports and see if some of these classic pro-inflammatory microbes are present and whether they might be changing the phenotypes that you see in the immune system or in any other system in your that you might be studying. That's the kind of practical take-home model message that I can. And if you do that and you find some interesting differences, it could lead to a Nature publication. So pay attention to it. But in a kind of broader scale for the human condition, I think it's really important for ALS and FTD, and also many of these neurodegenerative disorders like Alzheimer's disease, where they have familial cases and sporadic cases, to really take into consideration what the actual pathway is that gene that's mutated might be contributing to. Is it what cell types is it functioning in? Is it just functioning in the neurons? Could it be functioning in glial cell types? Could it be having a systemic effect in addition to a central nervous system effect? And how does the potential characterization of the contribution of the environment influence that gene's function in the pathways that it's important in?
Right. Thank you so much again for all the answers and this very nice message. And again, thank you all who joined for being here in this Public Journal Club session. And we hope we can we invite you to the next sessions that will be posted soon in Twitter. And thank you. See you next week, everyone. Thank you, Aaron, again.
Diego, can we take a picture?
Oh, sorry. So yeah, sorry, Aaron. If you can, everyone can turn on their cameras, and we should take a picture at the end of the Journal Club to see everyone. All right, so let me stop sharing my screen. Everyone say neuroinflammation. 1, 2, 3. All right, thank you so much, Aaron, again, and we hope we see you all next week with more
Thank
journals.
you
I
so
look
much.
forward to seeing you all in conferences in the future when we all come together again.
Thank you.
You.
Good one.